Hi, good evening to all of you. In continuation to my last video lecture, which was on transcription in bacterial system or prokaryotes, in this video lecture we will be talking about post-transcriptional modification, or you can say RNA processing in eukaryotes. This RNA processing occurs in eukaryotes, not in prokaryotes, because the prokaryotes, as we mentioned in our previous lecture, that in prokaryotes the genes are continuous and transcription and translations are coupled. So the RNA must be already ready-made. It will be ready for the synthesis. So no further change or modification could be done in the transcripts produced in case of prokaryotes. But in case of eukaryotes, as the genes are in the form of discontinuous, they are not continuous. They are split into exons and introns. The exons are the coding regions of the genes, and the introns are the non-coding sequences. Hello. If that transcript is directly subjected for translation, the non-coding sequences will also get incorporated into the protein, and the protein will become non-functional. So therefore the system allows it to, within the nucleus, it gives it time and space to split this part apart, to remove the intron regions, the non-coding sequences, and to join the exons, the coding sequences. Other than this, this is one type of post-transcriptional modification or RNA processing, and there is another kind that the two ends of the RNA that is made are also modified for the stability of the RNA or for the transport of the RNA. The 5' end gets a guanine residue, and that guanine residue is methylated at its 7th position. This is called 5' capping. On the other hand, the 3' end, it gets a poly-A nucleotide addition. This is called poly-A tailing or polyadenylation. Let us discuss all these things in details. As we mentioned, there are two types of post-transcriptional modification. The first one is splicing, you can say, and the second one is end modification. In fact, the end modification occurs prior to splicing, when the RNA synthesis has just ended up, when the transcript has just been formed. It is released when the RNA is released from the DNA. It is a heterogeneous nuclear RNA, hnRNA you can call it, or you can also call it premature mRNA, or simply pre-mRNA. And this RNA, if this is DNA, of course, if this gene contains say 3 introns, and say this one is exon 1, exon 2, exon 3, and it contains 2 introns in between, intron 1 and intron 2. These numbers are variable, of course, genes to genes. So suppose this gene contains 2 introns and 3 exons. So in the RNA also, the first synthesized RNA, it will contain these parts, all of these parts, the exons, exon 1, exon 2, exon 3, as well as intron 1, intron 2, and some 5' untranslated region on the upstream and 3' untranslated region on the downstream also. So this RNA is called the heterogeneous nuclear RNA. As soon as this RNA is being formed, the 5' end is already modified. It is modified. As soon as it is formed, the 5' end, it gets a G over here, and that guanine residue is methylated at its 7' position. So this is called 5' capping. This is called 5' capping in brief. And the 3' end also, it gets cleaved at certain positions. Say it gets cleaved. We will talk about that. Maybe it gets cleaved here, over here. How it gets cleaved, we will talk about that in details. And then where this cleavage occurs, after the cleavage site, polyadenylation occurs. About 80 to 200 adenine residues are added by an enzyme called polymerase, and then this hydroxyl group. This is 3' OH at the end. So this is called 3' polyadenylation. These two things occur as soon as the RNA is being formed, and afterward splicing occurs later. Splicing means removal of the intron regions. These regions will be removed, and the mRNA, the mature mRNA, will be synthesized. So if this region is removed, the RNA will be like this. We'll cleave this part. These are the introns. There are methods of removing them. I'm just showing it like this. So these exons will come and will join. This exon 1, exon 2, exon 3. And these introns will be spliced out. Intron 1, intron 2, they will be spliced out. These are UTRs, 5' UTR and 3' UTR. So finally the RNA will be shorter than its precursor, that is the heterogeneous nuclear RNA or pre-mRNA. Or you can also say with respect to the gene, the mRNA, the mature RNA that is being formed after this modification, it is comparatively very shorter. The intron parts are removed, and this was proved for the first time in 1977 by what is Hogness et al. They proved that when they hybridize this mRNA with the respective DNA, with its respective gene, that is double-stranded DNA, denatured double-stranded DNA, then what they saw that the DNA is, some part of the RNA is hybridized. So this is the RNA, and the DNA forms loops like this at certain regions. Okay, these loops indicate the intron regions. The number of loops will be same as the number of introns are present in that particular gene. So this experiment, for the first time in 1977, showed that the eukaryotic genes contain exons and introns. It is not continuous, it is discontinuous. And this concept, in fact, is known as a split gene concept. It's called split gene concept. So this diagram can be drawn to show the split gene concept, that how the gene is finally expressed in the RNA. Here the entire exon part and intron part, everything is present in the heterogeneous nuclear RNA, the first formed RNA. But later the size is not proportionate, that is, it should be drawn in proportion. Let me change a little this last one. Suppose it ends here. So in this diagram, what is happening, the intron part is being removed like this. So it is getting shorter and shorter again. Okay, no, not this. Sorry. This one will come here, and this will end here. And this is the end result of that. The intron parts are removed. Okay. So finally, the exons that come into the RNA are joined together. Exon 1, exon 2, exon 3. Exons are the coding regions, intron are the non-coding regions. So now the gene, the mRNA becomes continuous with the coding regions. The non-coding regions are removed, spliced out. This is what is splicing is. There are various types of mechanisms of splicing. We will see uh, about them shortly. So now let us talk first about end modification. Then we will go for splicing. End modification, and firstly we will talk about 5' capping. What is 5' capping? As we mentioned that that the hnRNA that is produced, it contains a triphosphate at the end, 5' end. The first step in 5' capping is the removal of the gamma phosphate, the last phosphate, that is the gamma phosphate. And this is catalyzed by the enzyme called phosphohydrolase. Okay, so the gamma phosphate is removed and the RNA is now with two phosphate molecules at the end. And in the meantime, another enzyme called the guanylyl transferase. This enzyme, guanylyl transferase, this enzyme introduces a guanine residue from GTP at this position, and now the RNA looks like it has a guanine monophosphate over here, and this is 5' end, of course. And then what happens? A methyl group is added at the 7' position of this guanine with the help of the enzyme called guanine 7-methyl transferase. So now the RNA will have a PPP guanine 5' and of course its 7' position is methylated now. This is what is called 5' capping. And sometimes what happens are the second nucleotide also, the second nucleotide which is present here at this position, it also might get methylated. So NP at its 2' position, 2O position, methylation also occurs in certain mRNA, not in all mRNA. So all these things, either a single guanine molecule or the second one, it gets methylated. Both of them, this mechanism, this process is called 5' capping. Okay, this is about the methyl group donor is a molecule called S-adenosyl-methionine (SAM), and it is converted into, after donating the methyl group, it is converted into S-adenosyl homocysteine residue. This GTP, after donating the GMP group, the pyrophosphate moiety is released over here. So this is how the 5' capping takes place. The 5' cap is the recognition site for the protease ribosome to bind here and to start scanning the process from 5' to 3' and it does a sort of scanning to find the first AUG sequence from where the translation will begin. Now let us talk about the second end modification, which is 3' polyadenylation. So 3' polyadenylation. This is a very complex process. So when the transcript is being formed, it is at the verge of termination. Transcription is at the verge of termination. Then what happens? 3' end has to be polyadenylated, and as soon as the transcript is being released from the DNA, from the transcription bubble, it is clipped. It is identified by certain factors known as two factors are very important in this case. The CPSF, cleavage and polyadenylation, stimulating factor, in case of AST, it is simply called CPF, cleavage and polyadenylation factor, and the other factor is CSTF. This is also CSTF, cleavage stimulating factor. So these two factors, they have to come and bind into the RNA, a particular sequence of RNA that acts as a cleavage site. That sequence is called a consensus sequence, which is present somewhere from where the RNA is to terminate. It is present just before that a sequence which is consensus. It is called AAU AAA. This is the recognition sequence, and when this signal comes in the way, it is determinant that cleavage will occur just about 10 to 30 base pairs downstream of it, somewhere here. And this is the cleavage site, and downstream of that there is another conserved sequence, that is a GC-rich sequence, which is also a consensus sequence. So this is the signal for cleavage to occur. The transcript is a little longer, and it is cleaved at this position. So what happens? The CPSF, these factors, there are many proteins in CPSF. It is a complex mixture of proteins that recognize this AAU-AAA sequence. So this is, it is called CPSF. It binds to this recognition site, and then it is followed by the binding of the another factor, that is a CSTF in this GC-rich region. Okay, and one protein YSF1, which is a part of this CPSF. It is a part of this CPSF. It recognizes, it is the endonuclease that clips this particular RNA over here. Its name is YSF1. So this YSF1 is the endonuclease that introduces the cleavage over there. And so the RNA is now shorter than the actual size. So it is now AAU, AAA. After that, about 10 or 30 nucleotides downstream, the cleavage has occurred here. As soon as the cleavage occurs, another enzyme called the polymerase, that comes and binds here, and it introduces a number of A's, about 80 to 200 residues of adenine is added at this 3' end, and this is polymerase. This polymerase, it adds adenine residue at the end. AAU, AAA, triple A, then this one, and then number of A residue, A, colon A. So this is about 80 to 200 residues long, and it ends with a hydroxyl group 3' OH, that is correct. So the 5' gets a guanine residue, that is methylated at 7' position. This is 5' capping, and this is called polyadenylation. It's the poly tail at its end, 3' end. This is how polyadenylation occurs. There are two other factors associated with this, and this is in fact this research is a continuous process. It is going on. A number of protein factors are found every day. So there are other two protein factors which are important are CF1, CF1A, sometimes called CF directly CF1 or CF2, or CF1A, CF1B. These two factors also get associated with the CPSF and CSTF. This factor is CSTF. So this is the machinery for polyadenylation, and polyadenylation occurs to increase the stability of the mRNA in the cytosol, because there are many exonucleases that might cleave the RNA from 3' end, and it is also said that it helps probably when the RNA is being transported from the nuclei to nucleus to the cytosol to the nuclear core complex, then certain part of RNA might be cleaved at the end. So this extended part protects the RNA from that cleavage also. When the RNA comes out of the nucleus, the 5' end faces the 5' end comes first. This is the nuclear core, and this is the RNA which is coming out of the nucleus to the cytosol. Suppose the end that faces the cytosol is the 5' end, and this is 3' end. A number of proteins get associated with this to transport it. So it is basically a ribonucleoprotein complex which comes out of the nucleus into the cytosol, where this is what is 3' end modification. Now let us talk about splicing. Splicing, as was mentioned, that it is a process of removal of introns and joining of shuffling of exons together. There are two types of splicing, in fact. Self-splicing and spliceosome mediated splicing. These two types, we are talking about a type of splicing which in fact is called cis splicing. In that sense, there are two types of splicing broadly: the cis splicing and trans-splicing. Trans-splicing are very rare. They occur in case of protozoal system, where a different part of RNA from a different chromosome altogether it comes and binds with the spliced leader sequence, forming a Y-like intermediate. And this kind of because the RNA that is spliced, the source of that are two different chromosomes, two different locations. That's why it is called trans-splicing. Versus splicing, because it is a single strand within which the splicing occurs. So self-splicing is basically, it means the RNA, the intron, it gets automatically spliced. It was first noticed in case of tetrahymena, a protozoa called tetrahymena thermophila. In this organism, Seck and Altman were the first to say, Seck and Altman, and that gave rise to the concept of RNA world also, from where the concept of RNA world arises, because these RNA are specifically capable of catalyzing their own cleavage. So they are basically ribozymes. So for this RNA world concept, Seck and Altman, they were coveted by the Nobel Prize in 1989. for this discovery. So in fact, self-splicing is also of two types, group 1 and group 2. In both the cases, there is no enzyme or no protein involved. The RNA itself has got its capacity to cleave itself and rejoin the exons on the both sides. So how this occurs? First, let us see group 1 splicing. The group 1 splicing, so this is exon 1 and this yellow part is exon, it is intron, and this is again exon 2. There are two exons and one intron in the middle. So this intron has to be removed. What happens in case of group 1 splicing is a free hydroxyl group that makes a nucleophilic attack. It has a lone pair of electrons. It makes a nucleophilic attack at this end of the 3' end of the exon. This is 5', this is 3' end of the exon, this is 5' end of the intron, this is 3' end of the intron, this is 5' end of the exon, second exon, this is 3' end of the second exon. So this GOH, a guanine hydroxyl group, the hydroxyl group makes a nucleophilic attack on the 3' end, or you can say on the 5' splice site, and it breaks the RNA over there like this, and this, the intron, it hangs like this, and the rest of the part, the exon, it is attached to the exon, of course, here. So this is actually at the 3' end of the previous exon. It makes a nucleophilic attack on this end, and the two exons are then joined together. Okay, 5', 3', releasing this intron. Intron is clipped over here. The intron is now released like this. It is a linear intron. It is not a lariat. Lariat is formed in case of group 2 intron splicing, and also in case of spliceosome mediated splicing. So this is how group 1 splicing occurs. Group 2 splicing requires an intramolecular hydroxyl group. So in case of group 2 splicing, let me do it over here. Time is being over. Okay, I'll make it. Now coming to the group 2 introns. Group 2 introns occur in case of mitochondrial genome in eukaryotes. So group 2 introns splicing, group 2 splicing, it occurs in mitochondrial genome. What happens in group 2 splicing also, there is no requirement of any enzyme, proteinase enzyme rather. The RNA itself acts as a catalyst like group 1 intron, but the difference lies in the sense that there is no external source of hydroxyl group is required. It is intramolecular hydroxyl group. This is the adenine adenosine residue over here, that is called branch site. The 2' hydroxyl group of it acts as a nucleophile, and this site is called branch site. As after the intron is removed, it forms a branched lariat, a loop-like structure with a branch portion like this. This A site is located about 18 to 40 base pairs from the 3' end of the intron. Okay, so this 2' hydroxyl group, the lone pair of electrons from it, these are exons, exon 1, exon 2, and this is intron, of course. So what happens at the first stage, this lone pair of electron, it makes a nucleophilic attack at the 3' end of the upstream exon. The upstream exon and the intron, they split apart like this. and the intron forms, it covalently binds with the A site. This end of the intron, it moves like this, it binds, because this the 2' hydroxyl group of A attacks on this 5' end of the intron. This is 5' end of the intron, 3' end of the exon. So the 5' end of the intron, it gets ligated to this side. This is 5' end of the intron, like this, and this is the downstream exon 2. Then what happens? This hydroxyl group, the lone pair of electron of this oxygen, it makes another nucleophilic attack at this end of the this juncture, this end of the intron, the 3' end of the intron and 5' end of the exon, downstream exon, and creates a cleavage over there. The two exons are thus united by a phosphodiester linkage, and the intron, this lariat intron branch lariat, it is removed. So this is branch lariat. This is how group 2 splicing occurs. This is stage 1, step 1, stage 2. More similar to the group 2 splicing is the spliceosome mediated splicing. The video has become too much lengthy. Now let me wrap it up. The last part, that is the spliceosome mediated splicing. Spliceosome mediated splicing is very much similar to group 2 splicing because it also contains a branch site, this one, the branch site A site, and also forms a branch lariat. We will see it later on. What happens in group 2 in spliceosome is, the difference is this, that this spliceosome mediated splicing requires a complex of proteins that are together called spliceosome. So basically it is mediated by protein, RNA protein complex, that is called spliceosome. Spliceosomes are made up of proteins called U1, U2, U4, U6, U5, like this. They are designated as U's. There are many other U forms also, but in this case it requires these many U's. And these U proteins are basically one type of ribonucleoproteins rather. In fact, one snRNA, small nuclear RNA, is associated with a protein forming a structure called snRNP. They are pronounced as SNPs. So these U1, U2, U4, U6, etc. are SNPs. They come and associate with these intron and exon regions, and they accomplish the process of splicing. Now let us see how it occurs. So in case of spliceosome mediated splicing and also in group 2 splicing, the intron contains at its 5' end a conserved sequence GU and its 3' end AG. First of all, the U1 snRNP comes and binds at this position at the 5' splice site, and then binds the U2 snRNP also at the branch site over here. This is U1 and U2. After the binding of these two, they come closer to each other, they interact with each other, and the intron is folded like this because of that. The A site, the 2' hydroxyl group of that, this is brought closer to the 5' splice site, and it makes the nucleophilic attack over here. When this is happening, then this is U1, this is U2. Then what happens? U5 is also recruited, and it binds with the AG site on the 3' downstream, this side. So this is U5, and also the U4 and U6 also come in and they associate over here. U4, U6. Then what happens? This hydroxyl group, the lone pair of it makes a nucleophilic attack on the 3' end of the intron over here, And in the next step, the U4 and U6 they are released. U4 and U6 are released, and this intron forms a branch lariat, like structure. Next is forms. I have to draw it like this. Sorry. One more diagram. This is the lariat, still not clipped. It is still associated with the axon on the downstream. Axon 2, axon 1. Now this hydroxyl group, it makes a nucleophilic attack. It was making a nucleophilic attack. Other proteins, this U5 is associated with this. What was the color of that? I don't know, remember now. This is probably different. U5 and U2 and U1. They're still associated with the lariat. U4 and U6 have been released. Now, after this nucleophilic attack, a cleavage occurs here, and in the next stage, the lariat, the branch lariat, along with the U1, U2, and U5, U2 over here, U1 over here, and U5 over here, they are released, and the two exons are thus joined together like this. So this is how splicing in case of spliceosome mediated splicing occurs. Thank you very much.